So we are opening the after lunch session, the zoology session, and I'm very honored and happy to invite to chair this session the Tel Aviv University Vice President for R&D, Professor Yoav Ennis. Please. I want a joke first.
Okay. Okay. Thank you all for coming here. It's a pleasure to see so many people, and it's a wonderful conference so far, so I hope that this session will stand up to the level that we have seen in the former ones. And the first speaker that we have today is Professor Noga Kronfeld-Schor, the head of the School of Zoology at Tel Aviv University, who is going to speak on, are you a morning or an evening person, the adaptive significance of chronotypes. So I'm inviting Noga, but one word is because clocks. So there is this guy that comes to a foreign city and his watch stops, just stops still. So he is looking around where to fix it, and he sees a store with a big sign of a clock over it. So he goes there and he says, could you fix my watch? And he says, no. Why not? Because I don't deal with watches. So what are you doing? I am a mohel, so someone who is doing circumcision. So he says, so why did you put up a watch for a sign? He says, what did you want me to put up?
So you know,
Please, Noga.
this is a reflection of my being stressed.
בגלל זה לא עשה מצגת קודם. רגע. אצלי זה... לא, זה מזהה. זה כאילו כבוי. זה מתחיל, אני חושבת. הנה, הנה. Okay, thank you very much. I'd like to start by thanking Mira for organizing this conference for the second time. Mira, where are you? I don't see you. She's out, okay. And for inviting me to speak for the second time. So last year I told you about research that started here at the Dead Sea area on these rocky deserts, looking at two species of spiny mice that you can see in the picture, the golden spiny mouse on my right, which is diurnally active, and the common spiny mouse on my left, which is nocturnally active. And I talked about how the research developed into a study of diabetes and depression in a diurnal animal model. And this study is already published. And today I want to start from the same point but taking you into a different direction. Again, looking at these two species of spiny mice, but this time looking at their circadian system and specifically on individual variation in their circadian system. So during the last 2 years, we all heard a lot about circadian system, mainly thanks to these three researchers who were awarded the Nobel Prize for their discoveries of the molecular clock mechanism controlling the circadian system. But one question is, why do we call them circadian system and not diurnal. Circadian rhythms and not diurnal rhythms. And to explain that, I have to first show you what we do when we study daily rhythms or circadian rhythms. And what we do is monitor any variable constantly for a long period of time. We can measure body temperature, clock gene expression, behavior, activity, using diverse systems, and what we get is a huge amount of data that we have to analyze. And if we plot these data, and this is only 10 days worth of data, we get a huge set of data that you can clearly see that there is a daily rhythm, but it's very difficult to say anything about this rhythm. So in order to show it more clearly visually, what we do is transfer the data into a special graph called Octogram. And what you see here is the same data. The middle one? Okay. You see the same data, same 10 days, but this time they are depicted one below the other. So this is 24 hours, and the days are depicted one below the other, 10 days. And you can now clearly see that activity started here and ended here. So if we look at a schematic representation of such an octogram, we see here a nocturnal rodent or a nocturnal animal that started its activity at the same time every day over a few days. And the reason why these rhythms are called circadian rhythms is that when we keep these animals under constant conditions, we still get a rhythm, but it's not exactly 24 hours. So what happens is, in this case, the rhythm is a little bit more than 24 hours. It starts a little bit later every day. And we call this free running conditions. The clock is not receiving any information from the environment, but it runs freely according to its own molecular mechanism. And the word circadian means circa is about and dian is a day. Now this finding raises two interesting questions. One is the circadian clock has developed about 3.5 billion years ago. So over 3.5 billion years of evolution, it didn't get really accurate. It still doesn't count exactly 24 hours, which means that this must have some selective advantage. The other thing is that we all live in a 24 hours environment, which means that this clock has to be synchronized to the environment on a daily basis. So if we look at a central oscillator and an output rhythm, and it's not exactly 24 hours, we have to get an environmental input, and this input is light. It synchronizes the central oscillator and aligns the rhythms to a 24 hours world. But it's not that simple. So the internal clock has an influence on the phenotype, the rhythmic phenotype of the individual. So there's a large diversity between species and within species in the length of the internal clock. And for example, you can see here an animal that has a shorter internal period, which is also called tau, and a longer internal period. And the length of the internal period actually determines if we will be morning chronotypes or evening chronotypes. So if you look at the distribution in the population, and these are real data from a paper by Roenberg and Martha Merrow, you can see that there is a nice distribution which is a little bit shifted to the right. The right is evening people. The majority of the population is in the center. And if we look at the extremes, so the evening people have a long free running period. About 2 out of 10 are evening periods. Our evening chronotypes, they wake up late, go to sleep after midnight, need an alarm to wake up, and function well in the early afternoon into the night. On the other side, we see the morning morning chronotypes, about 1 out of 10. They have a short free running period, they wake up early, go to sleep early, don't need an alarm clock to wake up, and function well in the morning. I can tell you that I'm an extreme morning person and my husband is an extreme evening person. So today I woke up at 6, even though I'm not jet lagged. I ran 5 kilometers, came back, had a shower, and then at 8 came to breakfast with all of you. But that's what happens to me every day, even on weekends. On the other hand, my husband is usually going to sleep much later. And the first time that I understood why we have a snooze on in the alarm clock was when I met my husband. So this phenotype is, as I said, related to the circadian system. And today we know that there's a central clock in the suprachiasmatic nucleus of the hypothalamus, but actually every cell in our body has its own circadian clock, and they're all synchronized to the central clock in the SCN. And we can actually, instead of taking animals or humans into constant conditions and measuring their free running period, and this is a free running period of humans, we can actually take a tissue sample or fibroblast sample, infect it with a reporter gene and measure the free running rhythm of the cells in culture. And you can see that these free running rhythms, which were extracted from cell cultures, are correlated with the chronotype, with the time that people wake up and go to sleep. So this field of circadian rhythms, and especially chronotypes, was studied quite extensively in humans because it's really easy to follow them. We know when they go to sleep. We can ask them questionnaires about their habits and determine their chronotype. But to find what is the chronotype of a free living organism, we need an advanced technology which will allow us to monitor their activity patterns and feeding patterns and whatever we want to monitor in the field over time, which is challenging, but we managed to do it. And what we do is we implant the spiny mice with RFIDs and with body temperature transmitters or data loggers. And we monitor their activity, their feeding pattern in the field. And the first thing we asked was, do they actually show different chronotypes in the population? And to do that, we used these 4 large field enclosures. Each one is 20 by 50 meters. They're located not far away from here. And we can release the animals which we trapped around here, and then monitor, for example, their feeding habits by putting a tray full with 2 liters of sand and 3 grams of seeds mixed up, and putting an antenna that logs the ID and the time of the individuals that was feeding on in this food patch. And we can also record its activity and body temperature in parallel. And what we found was that indeed they show chronotypes. So what you see here is 3 of the 4 enclosures. In this experiment, in the fourth enclosure, we had a viper, which killed our animals. What you see here, these are different individuals in the first, second, and third enclosure, and you can see that there is a clear order of entry into this food patch, and the individual that enters first here or here is always first, and this individual is always last. And also we could record encounters between individuals, and we recorded 225 encounters. 90% of them were ended within 2 seconds, which suggests that these were aggressive encounters. They don't forage together on the same food patch. So are these true chronotypes? We conducted the same experiment again. This time we have the 4 enclosures and both species, the common spiny mouse, which is active during the night, and the golden spiny mouse, which is active during the day. And you can see that in all of them we see clear chronotypes, and the early chronotypes eat early and the late chronotypes eat late. We collected all these animals and transferred them to Tel Aviv University, measured them under controlled conditions, and what we found was a significant correlation between the order of arrival to a food patch and the free-running period, which suggests that the order of arrival is determined by the internal clock, as we see in humans. But how does it affect the ecology of the species? So using the same system, Ophir Levy, who was my PhD student, is sitting here and now he's a faculty member and he's going to give a talk later on in this session, modeled the food intake of these spiny mice based on the time that they spend on the tray and the amount of time that other individuals spent on the same tray before. Using a diminishing returns model, because the amount of seeds is constant. They are mixed in 2 liters of sand, and as an individual is feeding on these seeds, the attack rate is decreasing over time. And so looking at the effect of when arriving early to a food patch could allow the first individual to eat as much food as he wanted, but the individuals that get in less could do one of 2 things. It could stay for longer time and work more to get the same amount of seeds, or it could eat less. And what we found was that it is eating less. So this is the order of entry to the food patches and the food consumption, which was calculated based on the model. And you can see that the individuals that got in first ate more than the individuals that got in less. And this is interesting because we saw that when we keep these animals in the laboratory and we food restrict them, the golden and the common spiny mice can actually reduce their energy expenditure even when we restrict them to 50% of their normal food intake. And they do that by reducing their body temperature and using torpor. So we had body temperatures of all these individuals and we checked, is there a variability in the use of torpor in these animals? And these are 2 examples, 24 hours. And you can see that this individual hardly used any torpor and kept its body temperature high, and this individual was active a relatively short period of time, and it was active later in day, and it was using torpor while it wasn't active, and its body temperature reduced in some days even lower than 24 degrees Celsius. So can we find a correlation between chronotype, food intake, and the use of torpor? So this is the effect of food consumption on daily time torpid, and you can see that the correlation is significant. And the effect of order of arrival to a food patch on daily time torpid, and the animals that arrived later were using much more torpor than the animals that arrived early. So does it have any adaptive effect? And this is just a preliminary result showing that individuals that use more torpor, here on the right, have a lower testis weight, which means that using torpor reduces their reproductive activity, and we expect that these individuals will be will have less offsprings. In another study, we looked at the effect of light pollution in different wavelengths on activity in both species of common and golden spiny mice. And surprisingly, when we reanalyzed the data, we found, again, the same clear order of arrival in common spiny mice, which were active during the night in the control enclosures. But when the enclosures were exposed to white light pollution, you see that the animals were active almost all night, and there was a large overlap between them, meaning that first we expect to have more aggressive interactions and competitions, and we assume that they were more vigilant and afraid of the white light. Surprisingly, we found an influence only on also on the common spiny mice, which are active during the day, so they weren't exposed to the light pollution. And again, you can see that we have a clear order of arrival in the control conditions, but when the white light was on during the night, all the animals were active at the same time in the beginning of the night, and this relates to other studies that we did, and I don't have time to go into, but it fits well our results on the effect of moonlight on activity patterns. What we are doing now is we've just finished sequencing the genome of the two species together with David Thielbert from Earlham Institute. We have a collaboration with Steve Brown, who developed the assay for measuring tau using fibroblast tissue cultures, and we're trying to, instead of trapping the animals, bringing them back to the lab and holding them for 2 weeks to measure their free running period, we'll just take a tissue sample and create a tissue culture, and we could tell the free ranging, the tau of the free ranging individuals. And the third thing we're doing now is we just purchased a high frequency ultrasound, a portable one, which you can take to the field, and using it measure the number of embryos that the females have and the testis size of free ranging individuals, and measure the effect of different treatments like light pollution or the order of arrival. So I'd like to end with that and just thank the students and collaborators who did most of the work that I presented, and the 3 funding agencies, and thank you for listening. Any questions? Yes.
I have a question about jet lag.
Yeah.
Organisms are resetting their biological clock.
Yeah.
I am. You said they're controlled by a central clock, and then all the cells that are in the body are resetting to the original, to the master clock.
Right.
Oh, so what is the lag time? How long does that take, and what is the physiological consequence?
So the central clock in the SCN can shift about half an hour a day, but it also depends if you're a morning or an evening person, because if you're delaying your clock, it's easier for evening persons because they tend to be late anyway, and if you're advancing the clock, it's easier for morning persons. But the SCN is synchronized by light, and it can move about, on average, half an hour a day. But the peripheral clocks receive information not only from the SCN, but also from other signals like feeding, insulin levels, glucose levels, body temperature, oxygen consumption. And one of the problems with jet lag is that not all the clocks in the body are synchronized. So you have a wonderful orchestra without a conductor, and you get, this is why we feel bad, because we eat at the wrong time, we sleep at the wrong time, we don't sleep as well. And,
I got it.
yes.
I seem to remember that there are good data that shows that the biological clock changes with age.
Yeah.
Specifically in younger folks, it is, as you have here.
I knew that someone will ask that. So.
Do we know what causes the shift in the clock?
The simple answer is no. We don't. We're actually conducting also epidemiological studies on these spiny mice as a model animal, monitoring their free running rhythms throughout their lifetime. But as you can see here, this is humans with age, and you can see that people start with a relatively short free running rhythm. They're more morning persons. And then as teenagers, the clock is becoming longer and longer, and teenagers go to sleep later and later. And then at around 21 years old, it shifts, and it starts to get shorter again. And another interesting observation is that women's clock is always earlier than male's clock.
As a geriatrician, I could think that the end of that may somewhat be influenced by inability to sleep.
So at the end is also influenced by melatonin secretion, which is not functioning as well in adults, and then it causes an inability to sleep well. But that's on top of the shortening of the free running period. Yes.
It seems intuitive that if you're too far off the 24 hours, it's not very healthy.
Yeah.
So if you're, let's say, on a 26-hour rhythm, would it statistically lead to being a less healthy person?
Yeah. So there's research. So if 26 is very extreme, and usually they can't even synchronize to 24 hours, and they free run. So there are reports on people that are free running and can't actually synchronize to a 24 hours cycle. But even if it's not that extreme, even if it's 25, it's a delayed sleep syndrome. There's also an advanced sleep syndrome, and these people usually develop diseases like depression and in many cases also type 2 diabetes. Any other questions? Thank you.